So hello class, welcome back to Hamaji lecture, and this time we are now in lesson 4, and that is all about the energy metabolism of the red blood cell together with the discussion of its membrane physiology, that is its membrane structure and function. Okay, so here we go. So we have first energy metabolism. So our red blood cells, basically they need energy for what? So these are the following need or use of energy of a red blood cell. Number one, it's really to promote the membrane shape. Okay, so as again review, the shape of our red blood cell is described as a biconcave disc, and in order to maintain that shape, it requires energy. So it does not just occur or happen on its own, no? Sige lang siya biconcave disc, but there is really energy spent in order to maintain that shape. Second, of course, we need, or the red blood cells need energy in order to allow the different enzymes to function and react inside the red blood cell. Since again, our red blood cell is devoid of organelles, it only relies on the different enzymes present in the cytoplasm in order for it to go about its metabolism. Okay? And third, our red blood cells also need energy in order for the movement of different ions. For our ions, the red blood cell allows the passive diffusion of anions. So bicarbonate, chloride, they just go in and out of the red blood cell. But for cations like sodium, potassium, calcium, and magnesium, these four ions are cations, and they are actually transported actively. So via active transport that is requiring energy. Okay? And of course, for the reduction of the most important component of the red blood cell, that is specifically iron. Sometimes as your red blood cells transport oxygen, the iron is oxidized from ferrous or Fe2+ to ferric, that is Fe3+. So in order for it to be reduced back to its ground state, or basically its reduced state, it requires energy. So you can also check your books. You have their different uses or functions of our red blood cell, or for the energy of a red blood cell. There, box 9-1. So again, maintenance of the intracellular cationic electrochemical gradient, for the maintenance of the phospholipid of the membrane, for as well as the skeletal protein plasticity, for the functional, again, ferrous hemoglobin, for protection of cell proteins, especially against oxidative denaturation, and for maintenance of glycolysis, synthesis of glutathione, needed also for reduction of iron, and mediation of nucleotide salvage. So these are all the specific processes that require energy inside a red blood cell. So I've just given you here the major ones, or the four basic requirements of energy by a red blood cell. Okay, so let's go now to the form of energy. It's actually, of course, in the form of ATP. So as we've discussed in biochem, adenosine triphosphate is a high energy phosphate source, and it provides the greatest reservoir of energy, especially inside the red blood cell. So ATP, if you recall, is generated by a process known as glycolysis. So the glucose that enters our red blood cells is the main source, or the one that provides energy. So it's broken down by different enzymes according to our glycolytic pathways before, and the net yield is, of course, energy in the form of ATP. So ATP could also occur in the form of ADP or AMP, but the highest energy source is ATP. Okay, so what about the membrane exchange of cations especially? So just take note, the purpose why we need pumps, no, to transfer or transport cations into and out of a red blood cell is for it to maintain what you call as a gradient. It's electrochemical gradient, okay, or gradient. So take note of the gradient. For intracellular, the highest cation must be potassium. Okay? So nubo lang dapat si sodium and calcium. And for the outside of the cell, the highest naman ng mga cations are sodium and calcium, and potassium is low outside of a red blood cell. So meaning, if there are these three ions, potassium should be pushed into the red blood cell, while if there are sodium and calcium inside the red blood cell, they must be pushed outside or forced outside of a red blood cell. Okay? So the maintenance of this electrochemical gradient or gradient consumes 15% of the ATP generated by glycolysis. And if our red blood cells lack energy, kung kulang ka sa glucose, no? So kulang ka energy, our RBCs will be deprived of the energy needed to maintain its membrane gradient. So the masulod, mabaliskad siya, no? Masulod na lang si sodium kasi calcium, wala na may mapagwa sa ila, and si potassium pwede ka go out naman of the red blood cell, wala na may ma-retain sa iya sa sulod. So may reversal. So sodium, where salt is, water follows. So sodium enters the cell, water follows, and the cell will swell and lyse. So mag-burst ang red blood cell if maguba ang membrane gradient. Okay? So that's why we need energy to maintain that. Okay, what about the common components inside the red blood cell that are prone to oxidation? So again, the major cytoplasmic component of the red blood cell is hemoglobin, and our hemoglobin contains two major components, heme and globin. Okay, for heme, it's made up of another two basic components, iron and protoporphyrin 9. Okay, let's focus on iron. Again, for your red blood cells to effectively transport oxygen, the iron should be in the reduced state or ferrous state. Okay, but after it delivers the oxygen, it becomes oxidized in return, so it transforms to ferric state or Fe3+. So that ferric iron must be reduced back to the ferrous state. No? So what do you call now the hemoglobin that carries a ferric state of iron? So you have there methemoglobin. Okay? So if it's normal nga hemoglobin ang ngalan niya, it should be iron in the ferrous state. If it has already the ferric form of iron, we call it methemoglobin, and methemoglobin has no oxygen carrying capacity. So kung ferric siya nga iron, hindi katapik si oxygen. That's why we need to reduce ferric iron back to the ferrous state. Okay? That's the first na prone to oxidation, iron. What about the globin side? Okay, for the globin side, it's the sulfhydryl group. Okay? Sulfhydryl, SH, mga thiol groups na may sulfur sa aton nga globin chains. Remember nyo man, again, biochem in our proteins. Okay? The sulfhydryl groups causes, again, the bending and the loops in our chain. Okay? So again, your hemoglobin has 4 globin chains, by pairs na sila. For adult hemoglobin, it's alpha 2, beta 2, for a total of 4 globin chains. The part naman of your globin chains prone to oxidation is the sulfhydryl group, and once sulfhydryl group is oxidized, it will precipitate on the surface or membrane of the red blood cell, and it will form in an inclusion known as the Heinz bodies. Okay? Heinz bodies are actually precipitated globin chains due to the oxidation, of course, of the sulfhydryl groups. Okay? So Heinz bodies, of course, are not normal inclusions. So they can cause damage to the red blood cell. Pwede na siya kagtun sang macrophage. So the red blood cell now is nabuhinan iya nga membrane. So we need to avoid this nga inclusion to form in a red blood cell. Okay, so let's go now to the main source of ATP or energy in a red blood cell. As I have said, it's glucose. That's why we need to eat, of course, carbohydrates every day for the GO. Diba? GO, carbohydrates, that's for energy. Because the main source of ATP na ginakwaan sa aton nga red blood cells is the glucose, the monosaccharide unit of carbohydrates. Okay? What about the disaccharides? Or for another naman nga monosaccharide, galactose. Galactose could also be utilized. If you recall, pwede siya ma-convert man to glucose to enter the glycolytic pathway. Okay? And also fructose and mannose na disaccharides. Okay? Disaccharides na monosaccharides also. But for disaccharides and oligosaccharides like your pentoses in your mga nucleic acids or pwede man si ribose, no? They are not metabolized or cannot be metabolized by our red blood cells. So main gid na source is glucose. So how does glucose enter the membrane of the red blood cell? It's through facilitated transport. Okay, it's not active, it does not require energy, it's actually passive lang na siya. But remember the three types of transport across the membrane. We have the passive, the facilitated, and the active. No? Facilitated does not require energy, but it requires a transport protein. So may protein in the membrane of the red blood cells that facilitates the entry of glucose. You call that GLUT1. Okay, you can check your book. It's GLUT-1. GLUT1 is the transport protein for the facilitated transport of glucose into the RBC membrane. Okay, so let's go now to the pathways on how glucose is broken down. Okay? Or metabolized. So let's review lang. We've discussed this in biochem. We have the aerobic and the anaerobic pathway. The major pathway is the anaerobic one, and that is called the Embden-Meyerhof pathway or EMP. The aerobic is called HMS, hexose monophosphate shunt, also known as PPP or pentose phosphate shunt. Okay, so Embden-Meyerhof pathway, an anaerobic pathway, does not require oxygen, and it's a major one. 90 to 95% of glucose is metabolized via this pathway, and the end product is lactic acid. That's why lactic acid is a waste product. Okay, and it must be transported back, of course, from your tissues to the lungs and to your kidneys or to your sweat for excretion. What about these? No? In your Embden-Meyerhof pathway, we have also diversion pathways or may siya mga component pathways within mismo sa dako ni nga pathway. We have the Rappaport or Lou-Behring shuttle or Lou-Behring shunt. And the purpose of this shuttle is to produce 2,3-biphosphoglycerate. Okay? The purpose of 2,3-BPG, we'll discuss this when we go to hemoglobin metabolism. Siya ni ang regulate sang pag-carry ni hemoglobin sa kay oxygen. Or modulates oxygen transport in the cell. Another diversion pathway is methemoglobin reductase pathway. It's also found within the pathway, and ang purpose naman ni is to produce NADH or nicotinamide adenine dinucleotide hydrogen, which is the reduced form of NAD+. Okay? The purpose naman here of NADH, it's the cofactor of the enzyme methemoglobin reductase. From its name, it reduces methemoglobin, or basically it reduces ferric iron back to ferrous iron, which is very important in our RBCs. Okay, the net yield again of EMP is 2 ATP. Actually, 4 ang na-produce, it consumes 2. So, for a net of 2 ATP molecules. Okay, so that is again the overall Embden-Meyerhof pathway, if you can still remember. From glucose, these are the intermediate products, and the last product is the lactic acid. You can break this down into 3 phases. Phase 1. There you go. So we have 4 enzymes needed, and from glucose, the intermediate product is G3P or glyceraldehyde 3-phosphate. So hexokinase is the first enzyme to act. It requires energy, forming G6P, and G6P is broken down or rearranged to F6P via phosphohexose isomerase. F6P is added another phosphate group to form fructose 1,6-phosphate via another important enzyme, phosphofructokinase. And this one, just like hexokinase, requires energy. Fructose 1,6-P naman is converted to glyceraldehyde 3-phosphate, no, via the enzyme aldolase. Okay, so if you check this in our textbook, we have the summary in table 9-2 for 9-1, I mean, for the first phase, the enzymes that are involved, the substrates, their substrates, and their corresponding products. Among these enzymes, the 2 most important ones are hexokinase and phosphofructokinase. Okay, next, second phase. So to continue, second phase naman is in the middle part. So we start here naman from G3P. G3P is converted. May duwa siya ka pathways. Pwede siya ka-continue sa dalom, no? Or pwede siya ma-change anay into dihydroxyacetone phosphate, which is broken back again to, converted back again to glyceraldehyde 3-phosphate. So, overall, may duwa na ka G3P nga ma-enter sa second phase. So, this one is removed, no, sang hydrogen, ma-dehydrogenate siya ni G3PD to form 1,3-DPG or DPG. And then, another enzyme acting is phosphoglycerate kinase to convert 1,3-BPG to 3-phosphoglycerate, forming actually it's 2 ATP molecules because the one is siya ka G3P ang process. Okay? And in here, you can see now our 2 diversions. From the second phase, we have the methemoglobin reductase pathway, where you can see that NAD here is involved in converting G3P to 1,3-diphosphoglycerate. Okay? So NAD is now reduced to NADH. NADH is the cofactor of methemoglobin reductase enzyme, which will convert the ferric ion or methemoglobin to the ferrous na iron. Okay? The second diversion is the Rappaport-Liouvering shuttle or pathway. In here, from 1,3-BPG, i-convert siya ni BPG mutase to 2,3-BPG. Take note ha, the new name is BPG. So, in this illustration, it's the old name, DPG, pero it's now BPG. So, 2,3-BPG, which can be used by our cells in modulating oxygen transport, or it can be broken down further back to 3-phosphoglycerate via the enzyme phosphatase. Okay? So, let's go naman back to the summary of the enzymes in your book. So, you have here naman G3PD, phosphoglycerate kinase, by phosphoglyceromutase, and by phosphoglycerate phosphatase. And the third phase. Okay? Third phase naman of our EMP. So, starting naman from 3-PG or 3-phosphoglycerate, okay, it's converted by phosphoglycerate mutase to 2-phosphoglycerate. Rearrange lang. Then, by enolase to phosphoenol pyruvate. And last enzyme to act is pyruvate kinase, converting PEP to pyruvate. Okay? So, in here, it generates another 2 molecules of ATP for a total naman of 4 ATP molecules. Okay? So, Okay, so summary naman of the enzymes, it's in your book. You have their monophosphoglycerol mutase, phosphopyruvate hydratase, or also known as enolase, and pyruvate kinase. Okay, so take note, overall, for the three phases, we have important enzymes again. You have hexokinase, phosphofructokinase, phosphoglycerate kinase, and mga pyruvate kinase. These are important because once your body cannot produce these enzymes, we actually have a disorder or abnormality of red blood cells, and that is called HNSHA. Especially if you are deficient in phosphoglycerate kinase and in pyruvate kinase, these are common causes of HNSHA, which stands for hereditary non-spherocytic hemolytic anemia. So it's an anemia where your red blood cells are hemolyzed, and it's hereditary due to the lack of the needed enzymes in the metabolic pathway, and it's non-spherocytic, meaning the red blood cells are not spherocytes. So it's here man in your book. Okay, the most common cause good is deficiency of pyruvate kinase. Okay, so let's continue. Next slide. So these are the rate limiting enzymes, are basically the important enzymes of our pathway, and they regulate the rate, no, ang kadasigon sa pag-breakdown kay glucose. Hexokinase in the first phase has the lowest activity of all the enzymes. It requires magnesium as a cofactor and other cofactors, and its activity increases with increase in hydrogen ion concentration. So, kung acidic ang pH, ganubo ang pH, di bala, pagkataas si hydrogen. Kung acidic ang pH, ma-enhance ang activity ni hexokinase. Second enzyme in the first phase is phosphofructokinase, which also requires magnesium, also regulated or affected by pH, inorganic phosphates, and ADP. Okay, so that's for your EMP, the major pathway of producing ATP from glucose. Okay, the minor one is HMS, hexose monophosphate shunt, or PPP. Now, aerobic naman siya. In the presence of oxygen, glucose can still be converted, no? But it's just a diversion actually of EMP. No, siya sa first phase, kag sa first phase, doon nag-divert siya. So, in the presence of oxygen. So, only 5 to 10% of the glucose is metabolized via this pathway. Although minor siya, it's still considered very important because it's needed also for the maintenance of our hemoglobin molecule. Okay, but in a different naman na mechanism. So the purpose of this pathway is to produce what you call as the reducing potential of the cell. When you say reducing potential, hindi siya ang reducing agent, siya lang ang factor na needed to produce the reducing agent. So the reducing potential here is NADPH, nicotinamide adenine dinucleotide phosphate hydrogen. Okay, we've mentioned also this in biochem. So NADPH is needed to produce now what you call as the reducing agent, and that is glutathione in the reduced form. So the symbol is GSH, glutathione hydrogen. Okay, so NADPH is actually the cofactor of an enzyme known as glutathione reductase. If NADH is the cofactor of methemoglobin reductase in the EMP pathway, NADPH naman niya is the cofactor of glutathione reductase in the HMS pathway. The purpose is to reduce the oxidized glutathione back to reduced glutathione. So, the NADC HMS. So, going back again, this is EMP, but as you can see here, may a diversion from the first phase. Okay, Okay, so that is HMS or HMP. So from G6P, dira ma-enter ang pinaka-important nga enzyme also. Aside from glutathione reductase, we also need G6PD, glucose 6-phosphate dehydrogenase, because it's the enzyme that produces NADPH. Okay, so NADPH again is the cofactor of glutathione reductase to reduce your oxidized glutathione back to glutathione. NADPH could also be a cofactor of methemoglobin reductase together with NADH to reduce man iron back to the normal ferrous state. Okay, another pathway here, 6-phosphogluconate. This time it's 6-phosphogluconate dehydrogenase naman, converting to pentose 5-phosphate. That's why we call this pathway as PPP, pentose phosphate pathway. And in here, generating again NADPH, acting as a cofactor of glutathione reductase, converting your glutathione oxidized to reduced, together naman with your glutathione peroxidase naman. This could lead naman to a production of oxidized glutathione. So, dapat may ma-reduce sa iya, that is glutathione reductase. Okay? So, that's the purpose of HMS again. And important enzymes involved are glutathione reductase and G6PD. The principal reducing agent is reduced glutathione. The reducing potential is NADPH. Okay? Okay. So, again, special mention, si G6PD. G6PD, again, is the enzyme needed to reduce NADP while oxidizing G6P to 6-phosphogluconate. So, it's the only enzyme that provides the means to generate NADPH. If you are deficient of this enzyme, you have, actually you call it G6PD deficiency, which is the most common human enzyme deficiency worldwide. So in our case, here in the Philippines, we also have several patients na may G6PD deficiency. It's not fatal, but there are cases or situations that these patients or persons should avoid, especially taking certain medications, food like fava beans, and dapat hindi sila stress gid a lot, no? Because the red blood cells are not capable of producing NADPH and of course reduced glutathione. The red blood cells are prone to oxidative damage or stress, which leads to hemolytic anemia. So kung hindi man maagapan or ma-permit sila maka-encounter sa mga bawal, so they will be anemic. Okay, the red blood cells will lyse. Okay, it's not capable of reducing iron, no, back to its normal state, and hindi man siya, so it's not capable of transporting oxygen effectively. Okay na, so those are the pathways of producing ATP from glucose inside the red blood cell. Again, we need, or very important gid ang mga enzymes enumerated in the pathway, since without them, they could lead to certain types of anemias. Okay, next we go to nucleotide metabolism. So, just dasig lang ni siya because, of course, we cannot discuss biochemistry of red blood cells without discussing nucleotides. Okay, you have the major form of energy, ATP, which contains, of course, your nucleotide, adenosine. Now, if you remembered in our nucleic acids. So, nucleotides overall cannot be synthesized afresh, or meaning hindi nita ka-produce gid from some brand new nga nucleotides. All the nucleotides in our bodies are actually recycled. So, dapat hindi kita madulaan sa mga nucleotides kay they, of course, are very much needed in producing energy inside our body. So, the largest pool, no, aside from amino acid pool, the largest pool actually in our body is the adenine nucleotide pool, made up of 88% ATP, 10% ADP, and 2% AMP. The major enzyme needed to maintain this pool is adenylate hexokinase. Okay? So, what about the enzymes needed naman to salvage or to recycle or save the nucleotides in our circulation? We have adenosine deaminase. Deaminase naman niya, of course, siya niya ang gapaguba sa aton nga for regulation lang of our nucleotide. If it's decreased in concentration, madamo si adenine nucleotides. If taas ang enzyme nga deaminase, ma-deplete naman si adenine nucleotides. It breaks down your nucleotides, okay, for regulation only. And adenosine phosphoribosyl transferase naman converts our adenine to the first form of our nucleotide, which is adenine monophosphate. And if you remember, sa atong mga nucleotides needed by our nucleic acids, we also have cytosine, cytidine monophosphate, guanosine monophosphate, and thymidine monophosphate. So for our young red blood cells, we use the adenine, okay, na nitrogenous base. Okay. So overall, regulation of the metabolism of our red blood cells. Again, we have important enzymes, hexokinase, phosphofructokinase. Just take note lang what affects their activity. Hexokinase is sensitive to G6P. If damo si G6P, manubo si hexokinase. No? And the other way around, kung nubo naman si G6P, matdamo or ma-increase ang activity ni hexokinase. Its product is 2,3-BPG in the second phase. And in the first phase. Okay, so hexokinase is also controlled by 2,3-BPG. Phosphofructokinase, na inhibited also by ATP. Okay, because again, these two enzymes require ATP, if you recall, and another is 2,3-BPG. And both are inhibited by acidic pH. G6PD inhibited naman by NADPH. Kung damo si NADPH, manubo, ma-decrease ang activity ni G6PD. Then you have 2,3-BPG in the Rappaport Bering shuttle. Synthesis is controlled again by BPG mutase and degradation by BPG phosphatase. And ATP again is regulated in equilibrium to the amounts of ADP and AMP. The most important enzyme needed for its regulation is adenylate kinase. So that's for the regulation of the major enzymes in our RBC metabolism. Okay, so that's it for energy metabolism. We're going now to our membrane physiology of our mature red blood cell. Okay, so again, for the shape of the red blood cell, it's biconcave disc. And why is it shaped like that? The major purpose is to allow it to deform. No? Para mag-deform siya inside the blood vessels and hindi siya mag-rupture. Okay? So basically, it's needed also for the transport of oxygen and carbon dioxide. Okay, since palaba siya, no? Kung maglakad to siya lapit sa mga cells or tissues, dasig lang siya, it's easier for it to release oxygen and easier for it to accept carbon dioxide. Okay, that is because, again, of the high surface area to volume ratio. So what do we mean by high? Okay, so high surface area to volume ratio. So basically, the normal volume, the range of the MCV of the red blood cell is 80 to 100 femtoliters, or average tabi 90 femtoliters. For 90 femtoliters na sphere, na bilogya, gamay lang ang surface area. But because gin-flatten siya and naging biconcave disc ang shape, it has an approximate na surface area of 140 square meters, which is about 40% more nga mas dako ang surface area sang sa ordinary lang na sphere. So meaning kay mas lapad ang surface niya, mas dasig or mas damo oxygen and carbon dioxide nga ma-transport. And of course, it allows it to flex or deform inside small blood vessels. Okay, so meaning it can flex or it can stretch undamaged up to 2.5 times more. So hindi siya dasig-dasig lang yan ma-rupture. Okay, so first, for the deformability of our red blood cells, the first given factor that contributes to its deformability is its biconcave disc shape. Second, not in the PowerPoint, but second lang emphasis that provides deformability to red blood cells is the cytoplasmic viscosity. Ang iya mismo nga hemoglobin concentration inside the cytoplasm contributes to its deformability. That's why it should have lang the average na hemoglobin concentration. If you remembered, we have the MCHC na index ranging from, pila to gani? 32 to 37, in some books is 33 to 38 grams per deciliter. The purpose is to maintain lang the correct amount of hemoglobin inside. Kaya kung tamaan naman, it could lead to viscous na cytoplasm, so hindi siya maka-deform or maka-flex properly. Okay? So MCHC is also needed. Okay, so that's the illustration of our red blood cell. And side view, it has two biconcavities. And the purpose again of the biconcavity is to allow it to stretch or flex without damage. Okay? Okay. So let's go now to the composition of the membrane of the red blood cell. Okay? For the functions first, the purpose is to maintain the shape and its deformability, to maintain its osmotic balance. Well, what's the purpose sang mga unod sa membrane? Number 1, para maging ma-maintain ang iya shape and deformability. Number 2, to maintain the osmotic balance inside and outside of the cell. Third is to act as a supporting skeletal system for different antigens and receptors on the surface. And 4 is to aid in the transportation of different ions and gases. So I've mentioned before, for anions, they are transported passively, or yes, we have bicarbonate and chloride, but for your cations, they need an active transport. So we have pumps for that. For gases, of course, oxygen, carbon dioxide can just easily diffuse in and out of the red blood cell. Okay? So we'll discuss it later when we go to the different naman ng mga proteins. Okay, so how do we study the membrane composition of the red blood cell? Of course, we remove first the major cytoplasmic component, which is hemoglobin. So ang appear na lang is the membrane, and we call that a ghost cell, with a membrane alone without the hemoglobin. So the structure of the membrane is actually trilaminar, the same with other membranes, made up of 3 organic molecules. The major one is the protein, 52%, lipid is 40%, and carbohydrates is very few, 8% only. Okay, so let's start with the lipid structure of our bilayer. By the way, let's go back again to RBC deformability. So what are the factors that contribute to its deformability? Number 1, it's biconcave disc na shape. Number 2 is cytoplasmic viscosity, or the density of hemoglobin inside the cytoplasm. And the third, we have the membrane. Ang membrane mismo contributes to its deformability. We call that membrane pliancy. Pliant, meaning ma-bend siya without breaking. We also call that elasticity. So membrane elasticity is the third factor that contributes to RBC deformability. So ano nang contributes elasticity? So of course, again, it's caused by the lipids, phospholipid bilayer, and also the proteins inside. So review for the lipid bilayer, you can check your books. So again, it's bilayer, so it has an outer layer and an inner layer. For the outer layer, again, it faces the plasma. The inner layer faces the cytosol of the cell. And in the inner layer, we have the membrane cytoskeleton. So, proteins naman niya that are found in the inner layer of the phospholipid bilayer. So, let's start first with the lipids, which comprise 42%. So, again, we have the two major ones, phospholipid and cholesterol. Okay? So, they predominate the lipid fraction. They are in equal proportions. And these are, again, your phospholipids, if you can still remember, in biochem. So, hindi na pag-review ang structure, but in your red blood cell membrane, we have 5. Phosphatidylcholine, phosphatidylethanolamine, phosphatidyl'inositol, phosphatidylserine, and sphingomyelin. PC, PE, PI, PS, and S. Okay, so for their distribution, it's actually asymmetrical. PE and PS are found in the inner layer. Phosphatidylethanolamine, phosphatidylserine are actually found in the inner membrane of the phospholipid bilayer. Phosphatidylcholine, phosphatidyl'inositol, and sphingomyelin are found in the outer layer. Okay, that's the distribution. Additional, phosphatidylserine is very important because it's the negatively charged na phospholipid. So, ga-flip ni siya from inner to outer layer in cases where your red blood cells will participate in coagulation. So, this will be discussed naman when we go to platelets. No, kaya your platelets, white blood cells, have the same structure of membrane sa ilang just like your red blood cell. So, phosphatidylserine is negatively charged. So, sometimes your phospholipids can flip from inner to outer layer, from outer to inner, basta mo ni siyang distribution. Kung sinong ara sa sagwa, sinong ara sa sugod. So, the enzymes responsible for this are flipase, flopase, kung mag-scramble, so scramblase. Okay? So, the lipids can be attached to carbohydrates for the external sa sagwa nga half, and we call them now glycolipids. For the glycolipids, they're actually carrying antigenic structures, especially those involved in blood typing. So you have ABH antigens for ABO blood group and Lewis antigen, which is also a major blood group. Okay? So what about cholesterol? Cholesterol naman is important in regulating the fluidity of the membrane. Again, it provides rigidity or structure so that hindi siya too fluid. Okay? Also responsible in maintaining the surface area to volume ratio as well as the permeability to electrolytes and non-electrolytes. So that's naman for cholesterol. Next, we go to the proteins. Proteins are the major component. So 52, correct? Or 50%, I mean. So it again contributes to the shape and deformability of the red blood cell, especially the inner proteins. They also comprise our enzyme systems for the pumps. No? Sodium, potassium, ATPase pump, calcium, magnesium, ATPase pump. Okay, receptors for different molecules are also protein in nature, and antigens are also protein in nature, which these two are attached to carbohydrates, so we call them now glycoproteins, and they are found on the outer surface of the red blood cell. So how to study the proteins? We can extract them from RBC membrane, then i-electrophorese siya via PAGE. PAGE stands for polyacrylamide gel electrophoresis using sodium dodecyl sulfate nga buffer. Afterwards, we color them or stain them with Kumasi blue. Okay? So if it contains naman carbohydrates like sialoglycoproteins, also known as glycophorenes, we use naman another dye for that. That's PAS. PAS stands for periodic acid shift to stain naman our glycoproteins. Okay, so what are now the different proteins on the RBC membrane? So we have again two types of proteins. We have the integral. Integral is also known as transmembranous because it spans the entire membrane. So, and the second are the skeletal, also known naman as peripheral proteins, because they're only found in the inner layer. So let's start with the integral proteins first. We've just mentioned it a while ago. It's glycophorenes, also known as sialoglycoproteins. So it's a protein with carbohydrate unit with a sialic acid unit. That's why you have their sialic acid. So glycophorin is an overall name. We have different types, A, B, C, and D. And sialic acid, as I've mentioned, provides a negative charge to our carbohydrate coat, which we call again as the glycocalyx of the cell. Okay? Since all blood cells are negatively charged because of the sialic acid, we call the phenomenon nag-repel sila each other as zeta potential. So that's why they do not aggregate, they do not coalesce inside our blood vessels. They just bump and then bounce from each other because of the repelling of their negative charges. Okay, glycophorenes, since they are also mga proteins na may carbohydrates, they actually carry the receptors for our different nga mga ligands, and they also comprise what you call as the M&N blood group. Okay, another blood group found on RBC surface. Okay, so that's the illustration. We have here glycophorin A, commonly bound to band 3. So this is also glycophorin A. And we have glycophorin C bound naman to protein 4.1. This dark circle na structure. Oh, sorry. Okay. So, as you can see here, it's integral. If this is the phospholipid bilayer, it penetrates. That's why we call them transmembranous. And you will know that it has a carbohydrate structure. Kaya mga root-like structures are actually carbohydrates. These are the proteins, carbohydrate. Okay? So, in the carbohydrate here is sialic acid for glycophorin, providing the negative charge. Another important integral protein is band 3. Okay, sounds familiar. We've just mentioned it in RBC hemolysis in your extravascular, okay, or macrophage mediated. So when your red blood cells are near the aging process, senescent na siya, band 3 will aggregate with each other, forming again your senescent antigen that will attract an antibody, immunoglobulin, and will mark it for macrophage ingestion. Okay, so ma-hemolyze na siya. Another function of band 3 is actually a channel for our anions. As I have said, anions are actually transported passively, but they do not just simply diffuse, they are facilitated. So first, itong facilitated transport is glucose via GLUT1. Then your anions are also facilitated via what you call as AE1. AE1 stands for anion exchange channel 1, which is actually band 3. So chloride and bicarbonate, the two major anions in your red blood cells, actually move in and out of the red blood cell via this channel. Okay? Another one pagig na facilitated is water. Water has a transport protein, you call that aquaporine. Aquaporine. So for our passive gidyang wala gidya the required transport protein, you have the gases, oxygen, and carbon dioxide. They really are just diffusing passively in and out of the red blood cell. Okay, another function of band 3 is the key site of attachment. It's a large protein. I'll show you later in the picture. It's the key site of attachment of the lipid bilayer to the peripheral or skeletal protein. And band 3 carries your ABO blood group and the II blood group. Okay, so as you can see here, blood ko gid si band 3. These are the carbohydrate nga comprise ng aton nga mga blood groups or receptors. It's attached to your peripheral proteins. In this case, ankyrin complex, and in here, spectrin. No? So, siya gid ang major key site of attachment, no? Of the lipid bilayer to the inner portion of the red blood cell. Okay, so that's horizontal interaction. No, vertical interaction, I mean. Okay, so next, for the complete list, so I've just mentioned the two major ones. That you have glycophorenes and your band 3. By the way, these are your functions of your integral proteins. They support the glycocalyx through glycosylation. They are sites of transport and adhesion, and they are the signaling receptors as well for your cell communication. So here are the lists in your books. Okay, so these are the complete list. This is the complete list of your integral or transmembrane proteins. So, aquaporine for your water, band 3, calcium ATPase pump, Duffy blood group, GLUT1 for glucose, the glycophorenes, and the minor ones, and another ATPase pump for sodium, potassium, transporter naman for different ions, and the blood groups KEL, KID, RH, and HN. Okay, so minor naman na. Next, we go to your peripheral or skeletal proteins. They are fewer in number but still very significant. Same, no? Ang ilang significance. Okay, so peripheral proteins, also known as skeletal or cytoskeletal proteins. Major function, of course, skeleton, it provides the shape as well as deformability. Siya din actually nahatag sa membrane elasticity. Okay, so the major one is pectin. Other minor ones are actin, protein 4.1, and cheiron, G3PD. Okay? So, since major gets is pectin, we'll emphasize it more. It comprises the skeletal mass of your red blood cell, 50 to 70% yun. It's a dimer made up of alpha chain and beta chain. It actually contributes again to membrane pliancy. And it's connected to our different nga mga integral proteins a while ago via this na mga skeletal proteins man that serve as attachments. Protein 4.1 and ancheiron. That's why these two we call as complexes. We have the ancheiron complex, that's band 3, ancheiron, and spectrin. We have the protein 4.1 complex binding naman glycophorin C together with spectrin and minor peripheral proteins. So here you go. First is protein 4.1 complex, so shiny. This is spectrin. Beta monomer and alpha monomer, and the same one here. Since this is protein 4.1, it attaches glycophorin C to your spectrin with minor addition of adenosine, protein 4.9, actin, tropomyosin, and tropomodulin. Okay, it's the first complex. Second complex is the ancheiron complex, which connects naman band 3 to spectrin, plus the participation of protein 4.2. Okay, so ancheiron complex, protein 4.1 complex. And the last. Okay, so we now go to osmotic balance and permeability. So again, since our membrane, it's selectively permeable, it only selects anong i-allow niya entry and ang iya nga i-allow to enter freely in and out. Okay? So, again, anions facilitated siya via the anion exchange channel. Water is also facilitated via aquaporine together with glucose, but your gases are passively diffused. But what about your cations? Again, they require energy. So that's why we have what you call as the pumps. Okay, sodium potassium ATPase pump, calcium magnesium ATPase pump. For sodium and potassium, these two are paired together again. Okay, they are monovalent, Na+, K+. Calcium and magnesium naman are divalent, Ca2+, Mg2+. Okay, so first for sodium and potassium, take note of the gradient, intra to extracellular na ratio. For sodium, it's 1:12. Intra to extra. Intra si 1, extra. Cellular si 12. So, diin siya mas damo outside the cell. Okay? So, sa sulod, few lang siya, sagwa, damo. While potassium, the intra to extracellular ratio, 25:1. Almost twice ang kadamuon niya kay sodium. And diin siya makitaan inside the cell. So, it must be maintained ang among nga ratios. That's why we need an electrochemical gradient known as your sodium-potassium ATPase pump. So, that requires, again, energy. So, what do you think will happen if the pumps are damaged or if there is lack of energy na hindi mag-function si pump? So, of course, maguba ang pump, then sodium will go or enter the cell, potassium may diffuse out or siyang pagwaon sang cell. Wala na may ga-maintain nga pump. And where sodium is, water follows, the cell again will swell and will rupture, and amo na siya ang lead to hemolysis. Okay? So, you can read there the sickle cell disease na discussion in the book. Okay? And, let's check in the book for the sickle cell. Okay, so later lang na si sickle cell disease. By the way, the term for the hemolysis due to the influx of cations into the cell followed by water is called colloid osmotic hemolysis. Okay, so ang reason na diya is because the pump is not functioning well. Okay, and next, another cation, atong calcium-magnesium ATPase pump. If we compare naman the two divalent cations, magnesium should be high inside the cell compared to calcium. So, magnesium, if you recall, is very important na cofactor in our EMP pathway. Okay? For calcium is also needed but very small lang in amounts. No? So, calcium is needed by our spectrin. If our bones na skeleton ta sa lawas require calcium, the same goes with red blood cells. The spectrin, which is the cytoskeleton of the red blood cell, also requires calcium. Okay, so the pump is called calcium ATPase cationic pump. It maintains the intracellular levels of calcium to 5 up to 10 micromoles per liter of red blood cells. And the important binding protein that controls the pump is called calmodulin. Sorry, na mismo sa name niya, it modulates calcium levels inside the red blood cells. Okay, so lastly, the sickle cell disease is a classic example wherein gakaguba ang permeability of the membrane to cations. So just take note lang, we'll discuss naman this when we go to hemolytic anemias and to hemoglobinopathies. No, kung ang imo nga hemoglobin niya is may sickle cell disease ka, your hemoglobin is an abnormal hemoglobin that once it encounters an oxidized environment, masikol na siya. So, ma-condense, forming do katalum na hemoglobin. So, what happens inside the red blood cells, if nagsikol na siya, hindi na siya regulate the entry of cations. So, all cations will go inside sodium, potassium, and especially calcium. And since where there is the cation, water will follow and the cell will swell and will lyse. That's why sickle cell disease is a type of hemolytic anemia. So, anemic ka because your cells are hemolyzed because of the influx of water. So, that's also what you call as colloid osmotic hemolysis. Okay na? So, I think that's the end of our lesson 4. It's a shorter nga chapter as compared to the rest. So, guys, our class, continue lang to study and, again, prepare for a quiz next meeting.